Okay, so today our goal is to get through at least part of chapter 15, but we're gonna finish up chapter 14 first. You have a quiz today. It's open till midnight. Try to remind you as many times as possible. What? It is it's chapter 12 and 13. Right? No, it's 13 and a little bit 14. Whatever we did since last Friday. It can be 13. Chapter 15 homework is posted. I don't remember what day the syllabus said it's gonna be due. Homework syllabus dates are pretty much just wrong all the time. So I'm making it a bonus homework. So you can come in to the test on Wednesday. That means if you have a 0 for homework, you can replace that 0, and if you don't have a 0 for homework, it's gonna be a bonus for you. Okay? So it's posted, it's there for you, and that's the last chapter that's gonna be on the test, and that's what we're talking about today. The test is on Wednesday. It's on chapters 11, 12, 13, 14, and 15. So whatever we don't finish today in terms of chapter 15, we will finish on Monday, and then we're going to spend the rest of the class time reviewing for the test because it's 5 chapters. And it's a lot of chapters. And you're supposed to be working on your pedigree. So I'm going to give you a minute or two more to work on the pedigree side of numbers. Okay, remembering what we talked about last time, which I don't think I left that slide up there, but thinking about first trying to figure out what pattern of inheritance is it, and then labeling what you know, and then working backwards from what you know to try to figure out what you don't know. We're going to use big G's and little G's because they're easy to tell apart. Okay, so everyone remember all the ones that are all filled in are affected by whatever the trait is, and then you've got to kind of work from there. But your first job is to figure out, is it a dominant trait or is it a recessive trait? Well, that would roll for it. I'll figure it out. Talk about this one. Because it is kind of a tricky one. Okay, we talked about on Wednesday that the first thing you need to figure out is the pattern of inheritance. The pattern of inheritance is either dominant or recessive, because right now all we're dealing with is autosomal pedigree. So autosomal meaning either carried on the normal chromosomes. So dominant or recessive. And if you look at this one, what is your first instinct? Because you see a lot of affected individuals, your first instinct might be that this is a dominant trait. Because look, it's in every generation, lots of people have it, but spoiler alert, it's not. How do we know that it's not? What is the one clue that always tells us it's a recessive trait? There's one thing in here that tells us every time it's recessive that you have to look for, Brandon.
When the child isn't affected, but the parents are. When one of the.
Try the other way.
Yeah, when the parents are affected, the child is.
That's what you just said. He said the same way again. Trista, help him. The child is affected, but the parents are not. So right here in the middle, we've got this one couple that does not have the trait, but their kid does. Okay, the only way that can happen is if it's recessive. So that's what we're going to start with. We're going to start with these three people, and we're going to know that this one has to be little g, because it's expressing a recessive trait. It has to be homozygous recessive. I can fill in every single individual that is affected now because I know it's a recessive trait. Okay, from there, I can do some detective backwards reasoning, inductive, deductive, I can never remember. I can do some reasoning that might be inductive or deductive. It's deductive, I think. Okay, if this child is affected and the parents are not, what do the parents have to be? They have to be heterozygous because they're carriers. So they have to be heterozygous, big G, little g, and I shouldn't have circled them so dramatically. So sometimes when we do a pedigree, we actually color in ones that are carriers halfway to show they're not affected, but they are carrying the trait. Okay, so we know that, what do we know about 15 and 16? They're other kids. If parents are big G, little g, both parents are big G, little g, these guys have the dominant phenotype. What do we know about them in terms of genotype? Yeah, we don't know is really the answer. They could be homozygous dominant or they could be heterozygous. We can't tell. So we have to write both big G, or big G, little g. Okay, let's stick to the bottom row because we're there. For this family, number 5 is little g. And number 6, we don't know yet, but we know that 13 is little g. So if 13 is little g, what does 6 have to be? Has to be heterozygous big G, little g, because this kid got 1 G from mom and 1 G from dad. So this dad has to have a G. So again, we can color in half because that one's a carrier. What do we know about 14? 14 has to have a big G, right? Because they don't have whatever this trait is. And they have to have what? They have to have a little g because that's the only thing they could have gotten from mom. So they're carriers. Okay, if we come over to this side of the tree with 3 and 4's kids, we know that 9 is a carrier, 10 has it. What do we know about 12? We know mom is this. So what does this kid have to be? Has to have a big G because she doesn't have it, and has to have a little g because that's all that mom could give. So that kid is also a carrier. What do we know about the dad? He has to have a big G, right? And what else does he have to have? Little g, I know because this kid is little g, little g. One little g from mom, the other one had to come from dad. Okay, coming back over to this family, what about number 7? What do we know about number 7? Has to have a, ooh, hello, let's make it bigger. Has to have a big G and has to have a little g because that's the only thing that dad could give. We know that mom has to have a big G. What else do we know about mom? Does she have to have a little g? We don't know. So she's one we can't determine based on this pedigree. Okay. Again, I know you're practicing these in lab. We're not going to do this one together, but tell me if it's dominant or recessive. Oh, yeah, sorry. So eyes are complicated. There's more than one gene that affect eye color. So yes, and are all like environment also has an effect on traits. So the expression of traits. When I was in high school, we did this genetic thing where we surveyed our family, and supposedly the way you cross your hands like this is, did you do this one for lab? I can't remember. Yeah, so like left over right is dominant, which is the right way to cross your hands, and then the wrong way to cross your hands is the recessive way with right is over left. So we did this in class and we're like, oh, cool. And then we went home and we surveyed our family. And my sister's like, oh, I do right over left. I'm like, okay, I must have heterozygous parents. And then my dad's like, oh, I do right over left. And I was like, okay, so your mom must do left over right. She's like, no, I do right over left. And I was like, there's something that somebody needs to tell me right now because I'm dominant and everyone else is recessive. It was very clear genetically that I'm related to all of my family. If you ever met them, you would know. And so I went back to school the next day. I was like, what do I do with this? My teacher's like, there's environmental factors too. I don't think you need to like determine, but like eye color is just really complex. So yes, it's possible that weird eye colors can show up after you haven't seen it for a while. What? Oh, okay. So this one, we're not going to do it together, but what are you going to tell me? Is it dominant or recessive? We know because we have unaffected parents that have an affected offspring. We have unaffected parents that have an affected offspring. So we know it has to be recessive. So you can do that one for practice, for fun. I don't know if you find these fun. I find them fun. I really enjoy this, and wait until we get to the next one. The next one that I gave you, we're not going to touch right now because it's going to get into, it's going to relate to what we're talking about in chapter 15. So hide the royal family of England and Spain and Russia for a while. And we're going to start chapter 15. So chapter 15 is talking about the chromosomal basis of inheritance. So chapter 14 was all about Mendel, just the idea of genes. So we've already presented the idea of chromosomes, and we've talked about mitosis, and this is what happens with chromosomes, and meiosis, and this is what happens with chromosomes. But we have to remember that Mendel didn't even know about chromosomes. So we're talking about the law of segregation and how the alleles separate during meiosis, and they're on these chromosomes and they get pulled apart, and he had no idea about any of that. So we're gonna go, he was like 1860s, we're gonna go back to the 1900s, and I'm actually gonna read you a story. So you can put down your pen and your phone for a while, and I'm gonna read you the story about Morgan. So chapter 14 was about Mendel, chapter 15 is about Thomas Morgan. And this is the story of T.H. Morgan's discoveries in the field of heredity in 1910. 3 old ceiling fans fought to move enough air around the stifling lecture hall, we don't have that problem, to make the late September day bearable. Flies lazily buzzed from desk to desk, from notebook to notebook. Few of the students in the packed room had enough energy to shoo them away. It was a cruel late summer New York heat wave in 1910. Just when eager students at Columbia University should be diving into their new classes, the heat had sapped any interest beyond seeking the cool shade of a tree and praying for a hint of a breeze. The biology lectures of T.H. Morgan, the 44-year-old lanky Kentuckian with a great booming voice and permanent scowl of a skeptic, didn't help any. He was famous for putting as little energy into his teaching as possible. All his energy and time he saved for his research. The subject of this class was how different species evolved, changed, and adapted over long centuries. It was an important concept for all biology students to learn.
Morgan paced and droned on. His deep, monotonous voice seemed to have blended perfectly with the buzzing of flies. In the heat, freshest few of the students were really listening. I can identify with him sometimes. Professor Morgan, isn't evolution really a simple matter of survival of the fittest? Asked one student, hoping for a simple, straightforward answer. What? Survival of the fittest? Morgan stopped in mid-stride, surprised by the interruption. In truth, his mind was also far from the lecture hall. As his mouth rambled on about the flaws and existing theories of evolution, Morgan's mind was on his latest experiment, commonly called the fly ring. T.H. Morgan scowled and boomed out in his deepest, most intimidating voice. Put your phones away! Oh, he didn't say that. Put a plate of various bacteria on the table. If there's food, they all eat and multiply. When the food is gone, they all die. Where's the survival? Where's the fittest? A second student bravely came to the aid of his friend. But Professor Morgan, didn't Gregor Mendel prove that all changes in any species, peas, porcupines, or people, are controlled by genes passed on from one generation to the next? Morgan scoffed and glared at the poor student, who began to sweat under his fierce gaze. Have you seen a gene? Do you know anyone who has seen a gene? No, you don't, because no one ever has. Therefore, I don't believe they exist. If Mendel's genes existed, why wouldn't you look just like your parents, from whom you got all your genes? Genes are a convenient myth to explain what fools do not understand. If you can't see it or demonstrate it in the lab, don't ever believe it. Remember, ideas are a dime a dozen. A third student joined in, thinking Morgan was at least more interesting and entertaining when agitated by persistent student questions. Then how do species change and evolve, Professor Morgan? Mutation, random mutation. Morgan's voice echoed off the overheated plaster walls, causing two napping students to bolt straight up, tumbling notebooks and pens noisily to the floor. If you had read the chapter on DeVries' work with primroses, you'd know that. Class dismissed. And Morgan stomped out of the hall, even though there were 20 minutes left in the period. An hour later, Morgan climbed to the second floor of Columbia University's biology building and down the long corridor to the door marked the fly room. Morgan's fly room laboratory was a small, messy room with the overpowering reek of rotten bananas. Two walls of the room were lined, floor to ceiling, with rows of corked glass milk bottles containing hundreds of thousands of tiny fruit flies. Swarms of escaped flies, cockroaches, and mice scurried between hanging bunches of bananas. The constant buzz of countless flies was hard to talk over. Morgan's two hardworking assistants, cocky but quick and accurate Calvin Bridges, and the quieter, steady Alfred Sturvant, were bent over microscopes along the lab's back wall. When you have classes of your own, always neglect your teaching, boys, bellowed Morgan as he threw open the door. Research is more important and your lectures will be boring anyway, just like mine. Morgan peered over the young man's shoulders as they painstakingly examined fly after fly in the newest generation. Any luck? Asked Morgan, hoping against hope that at long last they would have found one of the random mutations he was searching for. T.H. Morgan had decided long ago to dig to the bottom of the evolutionary process. Believing the answer lay in a long series of random mutations, Morgan set up a lab to identify mutations and then follow their effect over many generations of a species. He chose fruit flies to study for three reasons. First, they are only 1/4 inch long. Second, they lived their whole life happily on nothing but mashed banana. And third, they created a whole new generation in less than 2 weeks. Morgan could study almost 30 generations a year. For 2 disappointing years, generation after generation had been carefully tracked with no mutations at all. It had been a mind-numbing effort for Morgan and his assistants. Each month, thousands of new fruit flies had to be carefully examined under the microscope for mutations, one by one. Morgan sighed and turned back to gaze at several milk bottles housing the newest generation. Suddenly, Calvin Bridges gasped and slid back his chair. White-faced, he bolted to his feet. He was almost too excited to speak, his voice a tiny squeak. Found one? An electric shock sizzled through Morgan. He spun around with his boomingest voice. What? After looking at 25,000 normal fruit flies, it was all Bridges could say. Found one. In 2 long strides, Morgan was across the room and had his eyes glued to the microscope, and there it was. A male fruit fly with clear, white eyes instead of the normal deep red. They had found a mutation at last. But would this mutation spread into future generations to change and evolve the fruit fly species, as Morgan believed it would? The white-eyed male was carefully segregated in its own bottle and mated with the normal red-eyed female. Now Morgan had to suffer through 2 long weeks of waiting for a new generation. The September heat wave finally broke. October rolled in cool and wet. Morgan's lectures continued to bore students, but Morgan didn't care. His mind was on the fly room. Finally, the call came to his office from Calvin Bridges. The next generation had hatched.
Morgan rushed up to the second floor. All Morgan's hopes and beliefs rode on the color of these flies' eyes. If they were white, off-white, or even rose-colored, as he was sure they would be, then the random mutation Bridges found would have changed and evolved the species. That was the theory of evolution Bridges presented. That was the theory Morgan believed. Took 3 days to examine the 1,237 new flies. Each and every one had normal red eyes. J.H. Morgan was crushed. The mutation hadn't helped. It had disappeared. It hadn't affected the species at all. Morgan was wrong. By October 20th, with fall colors still blazing around the Columbia University campus, the second generation, the F2, grandchildren of the original white-eyed male were hatched. There were over 4,000 flies in this generation to be counted. When Morgan saw the results, his face paled and his knees turned rubbery. White-eyed flies were back. There were almost exactly 3 normal red-eyed flies to each white-eyed fly, 3 to 1. That was Mendel's ratio for the interaction of dominant and recessive trait. J.H. Morgan's own experiment had just proved himself wrong and Mendel's gene theory right. Over the next 2 years, more than 300,000 fruit flies were examined in the fly room. Morgan found that Mendel's theory of genes and the traits they carried consistently predicted the way characteristics appeared in each generation. Genes did exist, just as Gregor Mendel had claimed 40 years before. In trying to disprove the gene theory in his messy, smelly fly room, J.H. Morgan actually proved it to be true. But what he did with 200 pounds of leftover mashed bananas is another story. I think it's interesting to think about the research. Maybe you don't. I think it's interesting to think about the history of how things came to be, because you read 2 paragraphs in the textbook about this. This guy spent years of his life counting fruit fly eyes. Not next week in lab, but the week after. Is that Thanksgiving? No, it wasn't Thanksgiving. The week after, you're going to be looking at fruit flies, and you're going to look at them under the microscope, and you're going to look at the color of the eyes, and then you're going to smash their little heads. And run chromatography with their eyes to see the different pigments that are found in their eyes. And maybe you'll think about this a little bit. One of my former colleagues just had a sign on your door that said, you know you're a biologist when you find a fruit fly, you look at the color of the eyes. When I find fruit flies at home on bananas, I'm always looking like, did I get a white-eyed fly? They're very rare in nature, but we have found different mutations that we have been able to grow in the lab. So you will have white-eyed flies and red-eyed flies and scarlet-eyed flies and brown-eyed flies. And you'll talk about the complicated metabolic pathways and genes that make those possible. But that was the basis. His discovery was the basis of what we call a chromosomal basis of inheritance, because he discovered not only white-eyed flies as having a mutation, but when he found that mutation, so in his parental generation, he had a white-eyed fly that was male, which he crossed with a red-eyed fly, which was, oh, I did that backwards, sorry. He crossed a white-eyed male with a red-eyed female. And in everything we've talked about so far, that wasn't really important, which parent had which trait, but this did turn out to be important to him. So when he made the first cross, his F1 generation was all red, which is what you would think if red was dominant over white. But his thought was, oh, the mutation disappeared. But when he crossed these red-eyed F1 flies with each other, what he found in the F2 generation was he got red-eyed females. All the females were red-eyed, which the story didn't talk about that. And he got white-eyed males. And red-eyed males in a 1-to-1 ratio. So he started to understand that the gene for the eye color for fruit flies was carried on a specific unit, which became the chromosome. We studied fruit flies because they're small, they have a short generation time, and they have 4 sets of chromosomes. They have 3 sets of autosomal chromosomes, 3 pairs, and then 1 pair of sex chromosomes. Just like humans, they have the XX, XY sex determining chromosomes. So he recognized in thinking through this process that these genes must be carried on something, and that something was the chromosomes. So the genes are on the chromosomes, and the sex chromosomes end up with this unique pattern of inheritance. It didn't disprove Mendel at all. It just added to our understanding based on this discovery of things being carried on the sex chromosome. So when we talk about humans, there's our fruit flies, red eyes and white eyes.
When we talk about humans, sex determination in humans is determined by the 23rd pair of chromosomes, which are X and Y chromosomes. So that's a TEM micro, image of a microscope, from a TEM microscope of the two chromosomes. What do you notice about the X versus the Y chromosome? X is a lot bigger than Y. And that is true because the X chromosome carries, in humans, about 1,100 genes. The Y chromosome carries 78 genes. So slightly more of your DNA comes from your mom, if you're a male, than from your dad, because you've got the X chromosome from your mom, and it's a little bit bigger than the Y chromosome. So those 1,100 genes on the X chromosome, some of them are sex determining genes that would produce female characteristics, but not all of them are, obviously. The gene that controls the development of the ovaries is actually on chromosome number 1. So that's an idea of more than one gene affecting the phenotype. For males, the 78 genes code for about 25 proteins, and some of those proteins are only found in the testes. And there's one gene on the Y chromosome that will determine the formation of testes. Sex determination is not the same across all organisms. So you can see here's a couple other organisms, like grasshoppers. If you've ever thought, gosh, how does that grasshopper know to be a girl or a boy? You've not ever thought that. I get that. Okay, so in grasshoppers, some other insects, can I read my own handwriting? Cockroaches. Yep. It's not an XY system, it's an XO system. So if an organism has 2 X chromosomes, then it's a female. If it only has 1 X chromosome, it's a male. In chickens, so in birds and some fish as well. Oh, I really can't read my handwriting. I didn't do well there. It is the ZW system. So in chickens, they have 76 chromosomes plus the Z and the W if they have a Z and a W. So if they have 2 different sex chromosomes, that makes them female, whereas the males have 2 Zs, which is the same chromosome. So that's the opposite of the XY and XO system. And then in bees and ants, there are no specific sex chromosomes. If an organism is diploid, if it has the full set of chromosomes, if it develops from a fertilized egg, then it is female. And if it develops from an unfertilized egg, then it is a male. So the males have no fathers. What I think is interesting about this is these animals are often social animals, meaning they have a hierarchical structure within they have a society where people have different jobs. Bees have different jobs and ants have different jobs, some of which are determined by whether they're haploid or diploid. So sex can be determined in a bunch of different ways. In humans, we're going to look at some examples of how this causes patterns of inheritance to be different. Okay, when we're talking about genes that are carried on the sex chromosomes, this makes the patterns of inheritance different. So we have the simple, like, oh, if unaffected parents have an affected offspring, it's recessive. If not, it's probably dominant. So now we're going to make it a little more complicated because it can be X-linked recessive also. I can't think of any X-linked dominant traits. But that doesn't mean they're not there. It means I don't know them off the top of my head. Okay, so can someone tell me an example of an X-linked trait? You know, any X-linked traits? Is anyone in here red, green, colorblind? Are you?
A little bit.
A little bit? Anyone else? You're red, green colorblind? Okay, so we have two people in here that are red, green colorblind, and both of them are male. And both of you have grandfathers on your mother's side that are also red, green colorblind. Because it's an X-linked inheritance trait. That often skips generations because of that, and we're gonna talk about that. So X-linked inheritance is just referring to genes that are carried on the X chromosome. And because they're carried on the X chromosome, how many X chromosomes does the male have? 1. So how many genes does the male need to express the trait? 1. So they only have to have 1 gene. So we're, instead of just being homozygous or heterozygous, we're going to introduce the term hemizygous. So hemizygous meaning like sort of zygous. Because you only need 1 allele to express the trait. So this is you can express a trait with only having 1 allele. So it's much more common for males to have an X-linked trait, to express an X-linked trait. So we're going to look at some examples. We'll think of this as red-green color blindness. So when we write these, we write it like we did with incomplete dominance. You have to write the sex chromosomes in there too. Okay, you have to include the sex chromosomes when you make a Punnett square. So this is, we're going to do 3 crosses, and then we're going to look at the example I gave you. So this is showing you we've got a female. She's got 2 X chromosomes carrying the dominant gene, so she's homozygous dominant for color blindness. So is she colorblind? No, because color blindness is a recessive trait. I don't think I said that. In this case, we've got a male that is colorblind. So hemizygous for the trait only has 1 allele, but is expressing color blindness. So when we write these out, that means this male can pass on the X chromosome with the recessive gene or the Y chromosome to the offspring. And the mom can pass on the X chromosome with the dominant gene or the X chromosome with the dominant gene. Okay, so when you fill in this Punnett square, you have to include the sex chromosomes. So the mom's passing on the X big N, the dad's passing on the X little n. Mom's passing on X big N, dad's passing on Y. Mom is passing on X big N, dad is passing on little n. Mom is passing on big N, dad is passing on Y. So this is showing you the probability here. When we do the phenotype ratio here, we have to pay attention to male versus female. So there's really 4 possibilities. You can have a female who's affected. We're going to just say colorblind because that's shorter. Female who's not colorblind. We can have a male who's colorblind, or we can have a male who is not colorblind. So of these offspring, how many of the females are colorblind? Let's start with this. How many of these offspring are female? 2. So these 2 of them, how many of them are colorblind? 0, because in order to be colorblind, you would have to be this genotype as a female. A female has to inherit 2 alleles with the recessive, 2 X chromosomes with the recessive allele. So we have no colorblind females. How many, yes. And how many are not colorblind? 2. But these 2 are what we would call what? These 2 are both X big N, X little n. What are they? They're heterozygous, so they're going to be carriers. What does that mean for their sons? They might be colorblind. Okay, how many males are colorblind in this? Both our males are this. Are they colorblind? Nope. So we have no colorblind males and 2 not colorblind males. Okay, so of these parents, we have a colorblind dad and a not colorblind mom who's homozygous dominant. It's not possible for them to have a red-green colorblind child. There's different kinds of colorblindness. Okay, try this next one, where we have a mom who's a carrier and we have a dad who is not colorblind. So remember, you got to write the sex chromosomes on there when you figure out the gametes, and you got to put the sex chromosomes on there when you fill in the Punnett square.
Quietly talking. How many of these kids are gonna be colorblind? One, male or female?
Male, okay. Even though the mom is only a carrier, she could pass on a gene. The recessive gene. To this kid. So it's possible to have one colorblind child. How many of them are carriers? One. So this means for this couple, they have a 50% chance of having a colorblind male son, and they have a 50% chance of having a carrier colorblind carrier daughter. But it's also possible that their kids would not carry the gene at all. Okay, last possibility. What if you have a carrier mom and a colorblind dad? What's going to happen? In this case, we see dad can only pass on to a daughter the colorblindness gene, and mom can either pass the dominant allele, so no, not colorblind, or the recessive allele. So when we fill it in, what percentage of the daughters are gonna be colorblind? Of the daughters, 50% are gonna be colorblind, the other 50% are gonna be carriers for sure. And of the sons, how many are gonna be colorblind? 50% colorblind, 50% not colorblind. Can males carry the colorblindness gene without expressing the trait? No. Okay, so if you're a female and your dad is colorblind, you know you're a carrier. There's no possibility of you not being a carrier. If you're a female with a colorblind dad, you have to be a carrier. If you're a colorblind male, you can't pass that on to your kid. It's not possible for you to pass it on to a son, I should say. You could pass it on to a daughter if you married someone who's a carrier. That makes sense. So I taught at a school for 2 years where the art teacher was colorblind, and she was a female and she had a sister who was colorblind. And I was like, that is so cool. You never meet girls who are colorblind. And then she told me in high school, she didn't know she was colorblind until high school in biology class, and I don't know why she didn't change biology, but her life goal at that point had been to be a gemologist and to like, you know, do things with gems. But when she found out she was colorblind, she realized it was not gonna be possible for her because she wouldn't have the ability to distinguish between colors well enough to grade gems in the way that she would need to as a gemologist. So her life goal was shattered in biology class in high school by finding out that she was colorblind. But I thought it was really interesting that both she and her sister were colorblind, which means her dad was colorblind and her grandpa on her mom's side was colorblind.
So she told me that, I was like, oh, your grandpa's colorblind on your mom's side and your dad's colorblind too. She's like, yes. So she based her career and became an art teacher instead of that. Also, it's interesting because I would think color would be important for an art teacher, but not important enough that it ruined her whole life, just the gemologist part of her life. Some other examples of X-linked inheritance disorders, I guess. So we talked about red-green color blindness, which isn't like a life. It's not like a life-ending kind of thing. Another example is Duchenne muscular dystrophy, which is not a good thing. It's not, it's a progressive disorder where you're missing a muscle protein. So you have a progressive muscle weakening. This is not exactly, the next couple of days of stuff is not exactly like life affirming kind of things that we'll talk about. And then another one is hemophilia, one form of hemophilia. Hemophilia is an X-linked disorder. So that is what we're going to spend the rest of the time on. So now you can get out the sheet. Our last letter when they go. So this sheet, does anybody not get one? So on this sheet, you see a pedigree, and this one is going to have a different pattern of inheritance because it's a little more complicated. So if you look at the individuals who are affected, what do you notice about the individuals who are affected? Which sex do they have? So in this case, this is like a two-step identification of what is going on here. So this is a real pedigree. This isn't a made-up one. This is a pedigree that starts with Queen Victoria, who was Queen of England in the early 1900s. And she had a son, Leopold, who had hemophilia. Okay, so when you look at this and you're looking at patterns of inheritance to try to determine how something is inherited, we were looking at, do we have unaffected parents that have an affected offspring? You can still say that because we're still looking for if it's dominant recessive. But if you look at something like this and you'll notice how many affected females are on this pedigree. None. Okay. When you look at a pedigree like this and you see every single individual that's affected on this is a male, that's your clue to say, something is weird about this. We must be talking about something that's carried on the X chromosome because it's affecting males more significantly than it's affecting females. So when we talk about this pattern of inheritance and we look at these kinds of pedigrees, we're talking about an X-linked recessive pattern of inheritance. So again, once you know that, you're still going to approach it in the same way. And what does that mean? What are we going to start with? We start with what we know. So what do we know about all the males that are affected? Let's use H for hemophilia. What does their genotype have to be? But we can't just say small h because we know it's X-linked. We have to talk about the sex chromosome. So we need to say X little h y. And so that's where we start. And this is hard to write on, sorry, because it's super tiny. So all of the affected males are going to be X little h y. If they are X little h y, where did that X little h come from? Their mom or their dad? Had to come from their mom, so every single one of their mothers has to be X big H because they're not affected, X little h because they're a carrier. So every single female that has an offspring that has hemophilia has to be X big H, X little h. They're heterozygous. So we still talk about females in this case as being homozygous dominant, heterozygous, or homozygous recessive because they're still carrying two alleles. We just call the males hemizygous because they're only carrying one allele.
Okay, what do we know about Queen Victoria? Parents. She's a carrier. So we know that her parents, one of her parents had to carry it. Which one do you think carried it? If her dad had carried it, he would have hemophilia. He would express it. So we can guess that it was probably her mom that was a carrier and dad was not. What do we know about every single other male in the bottom part of this? If they're not affected, what do they have for a genotype? They have to have that big H gene because they're not affected and they have to be XY. So we're not going to fill in every single square, but just know that all the squares that are not filled in at all have to be X big H Y because they're not affected by hemophilia. And all the ones that are filled in have to be X little hy. And then what about all the rest of these females that are open? What would we know about them? We don't know about them. And again, we're not going to fill them all in, but all these ones that are circles like this, unless they have an affected offspring, we can't know what they are. So unless they have an affected offspring, we have to assume that they are homozygous dominant or heterozygous. There's no way for us to know the difference. Like this mom is a carrier, so she could have passed on the gene to this kid, to this daughter, but we don't know because this daughter didn't have any children with hemophilia. So we could guess that she might be this, but it's still possible that she was a carrier of it. So all the ones that don't have any offspring with hemophilia, we can't tell the difference from that. So this is actually a real pedigree. And you can see that there was a lot of intermarriage among the royalty of Europe. So this includes the Spanish royalty, the Russian royalty, the German royalty, and this is the current English royalty, including Prince Edward, who I think is Prince Charles's brother. I think this is Queen Elizabeth and Prince Philip. So it doesn't include the next two generations. I don't know if you're a royalty watcher, but one of these would be Charles, who has two kids who have other kids, none of whom have hemophilia. Okay, that's where we're going to stop. So we will do the last slide of 15 on Monday. What do you have to do this afternoon or this evening? You have a quiz. You need to take it.